Hello, my name's Emily Burns, and I'm an environmental scientist at the Personal Care Products Council located in Washington, D.C. Before I get started, I'd first like to extend a really big thank you for inviting me to speak today at the Sunscreen e-Summit. My talk today is going to be on sunscreens and coral reefs and providing a research update on what we know in this area. I'm going to be looking at studies that go out into the field and measure concentrations and how those compare with laboratory studies that look at the toxicity of UV filters, and then how we bring all this information together within an environmental risk assessment to understand the environmental safety of UV filters. Let's start by discussing where this environmental safety concern for corals and their exposure to sunscreens come from. So we think that there are two main pathways in which UV filters could reach coral in the environment. And the first is through wastewater. And what I mean by that is that you would put on a sunscreen and you would wash it off either in the shower or put your clothes in the laundry. And then that water would go down the drain to a wastewater treatment plant where it would be treated and then released to the environment. And there would be some UV filters that would make it through treatment and be released to the environment as well. Now, in the US, the majority of wastewater from wastewater treatment plants is released to the freshwater environment, but in coastal areas or island regions, it could be expected that that wastewater could be released to the marine environment and then it would disperse and then potentially then interact with coral reefs. Now, the second pathway, which we think is most likely the dominant pathway for coral exposure to UV filters, is called point exposure. And what I mean by that is that when you put sunscreen on at the beach and then you get directly into the water for some form of recreation, the sunscreen will wash off you to varying extents, and then it disperses into the water and then has the potential to interact with coral. So it's this concern, along with a couple laboratory studies, which I'm going to discuss in a little more detail later, that's led to precautionary bans or restrictions on certain UV filters. A good example of this would be octinoxate and oxybenzone. A couple years ago, they were banned in Hawaii. They were also banned in Palau and the U.S. Virgin Islands. And more recently, there's been discussions to ban avobenzone and octocrylene. And most recently, there have been proposals to expand this ban to cover all organic UV filters. So what I want to do today is take you through the available science and review it, critically review it, and discuss whether it supports these proposed bans or even the existing bans for these UV filters. So before we get into talking about the actual studies itself, it's first important to understand how we actually establish environmental safety of an ingredient. And to do this, we conduct an environmental risk assessment. Now, to explain environmental risk assessment, I'm going to take it back to the grandfather of toxicology, Paracelsus. And Paracelsus said that all things are poison and nothing is without poison. So, for example, water or Tylenol, it's solely the dose that determines a thing is not a poison. So it's actually how much you're exposed to that determines whether the thing is going to cause harm or not. And this is really the one of the founding principles that underlies risk assessment. So I like to use this example with actually drinking water to kind of explain what I mean when we're talking about an environmental risk assessment. So as we all know, drinking water is absolutely necessary to maintain adequate hydration. And if we don't get enough water and become dehydrated, we can have negative effects. And if we get dehydrated enough, we can even die. On the other hand, there is such a thing as drinking too much water, and that can lead to water poisoning, which also has negative effects that we wouldn't want to have. On the other hand, there are certain points in our lives where even if we're exposed to a healthy amount of water when we're a little older, when we're an infant, like a newborn, we can't have any water at all. So that would be what is called a more sensitive life stage, because if a newborn has water, it could lead to hyponatremia. So even though water is absolutely essential to maintain healthy life, there is a point in which you can have too much. And this is what the risk assessment is trying to do. It's trying to determine what is that point that's too much and what is the likelihood that you're going to exceed it. Of course, certain people might be more likely to exceed it. For example, marathon runners who drink a lot of water at once might have a higher risk level. So let's put this in the context of environmental risk assessment. So the US EPA defines environmental risk assessment as an evaluation of the likelihood that an adverse ecological effect is going to occur as the result of an exposure to a stressor. Now, using our water example, water is the stressor, exposure would be drinking it. The adverse effects would be either water poisoning or hyponatremia. And the risk or the likelihood that those effects are going to happen is how much you have, how much water you have. So if we take this example and apply it to our coral, the stressor would be a UV filter. Exposure would be how much of that UV filter the coral is exposed to. And the adverse effect would be something like growth, coral mortality, effects on reproduction. And then the risk is the likelihood that those effects will happen based on the amount of UV filter that coral is exposed to. Let's talk more specifically about what's needed to conduct a coral environmental risk assessment. So the environmental risk assessment is divided up into three parts. The first part is the environmental exposure assessment. And the simple way to think about this is how much of a UV filter is in the water near coral. And we would figure this out by looking at studies from the literature that actually take measurements from the water that are representative of that environment. The next stage is the hazard assessment. And what that boils down to is what level of a UV filter harms a coral. So if we think back to our previous slide, harm is that adverse ecological effect. So at what level does a UV filter cause coral to die or reduce their growth or to have impacts on their reproduction? And then in the third phase, we basically are comparing our environmental exposure assessment to our hazard assessment to answer the question, is there enough UV filter in the environment to actually cause harm to coral or to cause that adverse effect? So let's talk about the environmental exposure assessment. So here I have a hypothetical beach shoreline scenario where the brown is the beach and the sand, the blue is the overlying water, and then we have our coral just offshore there in the little box. So someone on the beach uses their sunscreen and then they get in the water for a swim. We'll call that recreational wash off. So the UV filter washes off them and then is subject to a process called environmental fate. So the environmental fate of the UV filter is represented by that gray arrow in the water. And you can see that it starts off quite wide directly at the site of the wash-off and then gets narrower as it moves away from that wash-off site. And what that represents are the fate processes that could be acting on the UV filter. So the first one could be dilution, so it's just dispersing in the water column. The second, it could be degraded by the microbial community present. And the third, it could be partitioning out to the sediment because these UV filters are quite sticky and they actually really like to go to sediment rather than hang out in the water column itself. So then when it comes to the coral exposure, you actually have quite a bit less that makes it to those coral compared to where it was initially washed off. And so when we're looking at our monitoring studies, we're limiting the scope to those studies that are targeting samples near coral reefs, because this is actually the most representative of coral exposure. Because as you can imagine, before those UV filter fate, environmental fate processes kick in, it's quite a different level of UV filter directly at that wash-off site versus what actually gets to the coral. Now that we've discussed limiting the scope of monitoring studies to those that actually took samples near coral reef, how much is found where globally? So here is a summary of the studies that are currently available. In the Pacific, we have 2 studies from Hawaii, and we have 3 studies from Hong Kong. We have another 2 studies from Taiwan, and a final study from Japan, so making up that East Asia region. In the Atlantic, we have 2 studies that looked at samples from the United States Virgin Islands. We have a study that took a few samples in Bonaire, and also another study that took a couple samples in Grenada. So only 4 studies comparatively in the Atlantic. So overall, there's actually not that many studies that have conducted monitoring near coral reefs globally. So let's start looking at data, and we're going to begin with oxybenzone monitoring data. Oxybenzone has more monitoring data than any of the other UV filters, and as you can see in the plot below, I've divided the data up by region so it's a little easier to digest. So on the left, we have the studies from the Caribbean, in the middle, the 2 from Hawaii, and on the right, those studies that we identified from East Asia. And so here I've presented a box and whisker plot. And in the upper right-hand corner, I've included a little legend of what those boxes and those whiskers actually represent. And what I'm most interested in from a risk assessment perspective is the median value from each of the studies, so that line in the middle of the box. And I'm most interested in this value because it is the most representative of the variability across their study. It represents the midpoint in the data set, so it kind of gives us a good idea of what they were looking at. Because as you can see, some of the bars do vary quite a bit, which means that they had quite a variability in the concentrations they detected. That could be for a number of reasons. For example, they could have sampled at different times of day, they could have been sampling in different areas, or maybe they only sampled one site. So that's all going to be impacting how these bars look within this. And what I mainly want to draw your attention to are these 3 studies within the red circle. So in the middle, you have this Downs 2016 box, and it extends higher than any other study, and it has the whisker coming off. The maximum value is far higher than any other study that has been detected. So that's actually about a million nanograms per liter, which, bear in mind, this is also on a log scale, so small changes are much larger than you might think. So 1 million nanograms per liter translates to 1 milligram per liter, and we do know from studies of coral reefs that the dissolved total carbon content in a reef environment is roughly 1 milligram per liter. So oxybenzone would fit in this category of dissolved organic carbon because of its chemical structure. And so what this data point is saying is, hey, there's as much oxybenzone as there is total dissolved organic carbon in the reef environment, which would indicate that it's completely swamping out the other sources of organic carbon and potential. It seems a bit off. So that's what we would call something like a sanity check. It doesn't quite seem right. And when we looked at the methods behind their study, we found they were quite outdated. Their detection limits were quite high, so they couldn't detect very low concentrations. In fact, their detection limits were higher than the maximum concentrations found in several of the other studies. And on top of having kind of poor method sensitivity, they were also missing a lot of key components of best practice in terms of quality control. And when we looked across all the studies, we did find this was a recurring theme. There were important elements of quality control and assurance that were missing, or the ways in which they quantified their concentrations were not quite in line with best practice for environmental monitoring data. But looking at the median across the studies, so the line in the middle of the box, what we find in general is that it sits between the 10 and the 100 nanograms per liter. And so that's our big takeaway, is that on average, concentrations seem to be sitting between the 10 and the 100 nanograms per liter for oxybenzone in a near reef environment. So how do the monitoring data from the other UV filters compare with oxybenzone? As I mentioned, there's not as much data for the other UV filters available as compared to oxybenzone, but I think there's still some key takeaways for each of the UV filters that I'm going to try and walk you through. So first, starting with octinoxate, let's focus on mainly the Hawaii data here, this Mitchell-Moore 2019 data. The detection frequency for octinoxate in their study was only 16%. That's much lower than it was for oxybenzone. And what that's telling us is that while it's there, it's not there as often, it's not there as consistently. And also the concentration in which they detected is much lower than for oxybenzone. It's about 1 nanogram per liter. And this is reflected across the other studies that have looked at octinoxate. The concentrations do seem to be quite a bit less than they were for oxybenzone. Now for octocrylene, focusing again on the Mitchell-Moore study in Hawaii, you have a 95% detection frequency. So in comparison to octinoxate, it's detected much more frequently. So it is present. However, in comparison to oxybenzone, the concentrations are quite a bit lower. So the midpoint is sitting between 1 and 10 nanograms per liter rather than 10 and 100 nanograms per liter. And again, this is similar across the other studies that we have found for octocrylene. And now finally, avobenzone. Avobenzone has the least data available for any of the UV filters. Mitchell-Moore et al. Looked for avobenzone, but they were not able to detect it in a single sample. So we don't have any data from avobenzone from Hawaii or the Caribbean. We do have 2 studies that were able to detect avobenzone from Hong Kong, but that's about it. Okay, so next we have data for homosalate, and we have data from each of the 3 regions for it, but again, I'm going to focus first on Hawaii. So there's a 68% detection frequency from that Mitchell-Moore et al. Study, so a little less than oxybenzone, but pretty regularly detected. What's also interesting is the level at which it's been detected. So instead of that median value or the midline in the box falling between 10 and 100, it seems to be just a little bit higher than 100, which would indicate that homosalate concentrations seem to be a little bit higher than oxybenzone. On the other hand, octosalate only has data from Hawaii and East Asia, and the detection frequencies seem to be quite a bit lower for octosalate compared to homosalate. So for Hawaii, for example, there's only a 31% detection frequency. In addition, the concentrations do seem to be on average a bit lower as well. They're falling between that 10 and 100 nanograms per liter rather than exceeding it. So what does all this data mean? The first thing, we can see that there's quite a bit of variability within the studies, as well as between them and between the regions. So the UV filters were detected mainly at low nanogram per liter levels, so those median lines were either between 1 and 10 nanograms per liter or between 10 and 100 nanograms per liter. But when we take a step back and look at all of the concentrations reported, all of the bars, the majority are less than 1,000 nanograms per liter, which means that they're less than 1 microgram per liter. And I just want you to remember that 1 microgram per liter because it's going to be important when we start to talk about the toxicity data. So to put what this 1 microgram per liter in context actually means, it means about 1 drop in 1 billion drops of water, or about 1 drop of water in an Olympic-sized swimming pool. So that's quite a small amount. And to think that the vast majority of measurements are far below that. And when we think about the median values are much further below that again. The next phase of our environmental safety assessment is the hazard assessment. So here I have a hypothetical laboratory exposure for coral. And at the top I have an adult coral and at the bottom we have a larval coral. So the test could be based on either of these 2 life stages. And if you remember back to the environmental risk assessment example I gave with humans and water, do you remember how there were more sensitive life stages, for example, the baby versus the adult? So the same thing could happen in the environment. So it's important to test different life stages to make sure that we're basing our risk assessment around the more sensitive life stage. It's not always larvae, sometimes it's the adult, so it's important to get data for both. And in this case, we'll put the coral basically in the test vessel here and fill it with the appropriate amount of exposure medium. That would be some sort of salt water with the appropriate nutrients. And then you would add in your stressor, or in this case, oxybenzone. And then to run your test, you're going to have a series of treatments. And what I mean by that is different levels of oxybenzone in order to determine which level causes the effect. And within each of those treatments, you're going to have a series of replicates. And those are really important to get right. You need to have enough to make sure that the effect you're seeing is real. And when you're doing your statistics, it's really important to have enough replicates to show that. And for each of these little coral vessels that are included in this 96-hour hypothetical study, you're going to look at a series of endpoints. In this case, I've chosen mortality, polyp retraction, growth, and bleaching. And you're going to record the amount of effect at each of those treatment levels. And you're going to use that to determine and use statistics to determine what your final effect is for each of those endpoints. So for risk assessment, that's the no observed effect level, or NOEC, that we're most interested in. And so I've kind of written out what the hypothetical NOEC would be for each of these endpoints based on the data provided in those graphs. So when we're looking at these laboratory studies, it's this NOEC, that we're going to be looking for. That's the important piece of information that we really need to use in our risk assessment. So what studies are out there? Now, we found 2 studies that were looking at the larval stage, and then we found a further 9 studies that are looking at the adult stage. And when we look at the species that have been studied per endpoint, we find that the most commonly studied species is P. demicornis at 34%, followed by S. caliendrum at 30%. And this can be explained by 2 studies in particular, this Hay et al. 2019 A and B study, where they looked at multiple different UV filters and endpoints, and these were their test species. But overall, there have been several different coral species that have been studied in these test systems. Now, in terms of the UV filters that have been studied to date, the most commonly studied is oxybenzone at 31%, and that's followed by octocrylene at 12%, and then you have benzophenone 1, 8, and 4 just behind that. And again, that's due to that Hay et al. Study. On the other hand, you have avobenzone, octosalate, and homosalate being studied comparatively very little. The next important piece of information to look at are the endpoints themselves. And when we summarize them in this pie chart, we found that bleaching accounted for 24% of the endpoints studied, followed by mortality at 20%, algal density, which is a measure of bleaching, at 19%, followed by polyp retraction at 11%. Now, it's important to understand that not all these endpoints are created equal. Not all of them are ecologically relevant. An endpoint needs to be ecologically relevant to be used in an environmental risk assessment. So generally, endpoints related to mortality, reproduction, and growth are considered to be ecologically relevant. To be an ecologically relevant endpoint, it needs to have a demonstrated population level effect. And this has been done for coral in Australia when they were creating their guidelines for fresh marine water quality. So out of the endpoints that have been studied, I've highlighted in the green boxes which ones would actually be ecologically relevant. That would be bleaching, mortality, algal density, because it is a measure of bleaching, settlement, which is where larvae settle on a surface and then are able to grow, so that's related to reproduction, and then growth itself. Of the studies to date, only 68% of the endpoints that have been reported would be considered ecologically relevant. Now, taking a step further, there's also the concentration in which these endpoints were studied to be considered. So, for example, all the UV filters have a point in which they're no longer soluble, a concentration in which they will precipitate out of solution, and therefore it would not be realistic. It wouldn't happen in the environment to have a concentration above this point. So when UV filters are tested above this point, it's also not considered to be environmentally relevant. So we found that only 45% of the endpoints were actually tested below UV filter solubility. And you might be wondering how it's possible to test above UV filter solubility, and there are aids you can use in your test system to make things a little more soluble. And when you take both ecological relevance and solubility together, we found that only 37% of the endpoints reported to date are both below the solubility for the specific UV filter and also ecologically relevant. So I wanted to summarize all of the toxicity endpoints for you, regardless of whether they were ecologically relevant or not, so we can get a good look at how they all fit together and how they all look. So what I've presented here is called a cumulative endpoint distribution, and it's basically a way of just looking at the distribution of NOx, so those no effect concentrations, for each of the endpoints to kind of see how the different endpoints compare, how the different UV filter sensitivities compare, and how the different species sensitivities compare. So there's a ton of data here. I don't expect you to take it all in. I'm going to walk you through some of the main points here. So on the x-axis along the bottom, that's the exposure concentration. So for example, you have polyp retraction. That NOEC, or no observed effect concentration, for those UV filters is at 1 microgram per liter, and that would be our most sensitive endpoint. And then we jump up to 10 micrograms per liter, and then 100, 1,000, and 10,000. And so inside this red circle, we actually have our lowest ecologically relevant endpoints, and those are for benzophenone 8. And then we have oxybenzone and octinoxate at 100 micrograms per liter. They have mortality and bleaching endpoints there. And then octocrylene is actually our next lowest ecologically relevant endpoint here at 1,000 micrograms per liter. So there's about a factor of 10 between each of those. And the main takeaway here is that, yes, effects have been observed down to low microgram per liter levels. If you remember back to the exposure data I was showing you, remember how I said most, like the vast majority of the data was actually below 1 microgram per liter. So just keep that comparison in mind. The most sensitive endpoint is polyp retraction, but as I said, that's actually not an ecologically relevant endpoint. On the other hand, you do have mortality, bleaching, and algal density there at 10 micrograms per liter, which is relevant. On the other hand, there's no ecologically relevant endpoints available yet for avobenzone, homosalate, and octosalate. I've talked a bit about how an endpoint might not be relevant for an environmental risk assessment, but it's also important to evaluate whether an endpoint is reliable for an environmental risk assessment. So government organizations, they actually evaluate data quality prior to using an endpoint in an ERA, or environmental risk assessment. And this is because you want to be basing your ERA on robust, reliable data, because it's going to be used in environmental decision making. And the quality of your decision making is really going to depend on the quality of data you're using to make it. So in the US, you have the Information Quality Act, which gave the US EPA a mandate in order to develop this quality criteria that a study must meet in order to be used in their decision making. And other regulatory agencies globally have developed variations of these types of methods. For example, Environment and Climate Change Canada have a method that was developed in 2009 by Breton et al. The European Chemicals Agency uses a slightly different approach. It was developed by Mormon et al. In 2016. But the main takeaway is that data reliability assessment is conducted to make sure data are actually suitable to be used in an ERA. So we've actually conducted a reliability assessment for each of these coral toxicity studies. If you're interested in reading more about it, I direct you to the paper cited at the bottom. Our assessment was a 23 really comprehensive data quality assessment, and it was based on that US EPA guidance, the guidance that I mentioned from Canada, as well as the European Union, to really give a comprehensive and holistic look at all of the aspects that are important to doing a good study. And they focused on these 5 criteria that I talked about: the test setup, compound, organism, test design, results, and statistics. As you can see, the majority of questions, 10 questions, were focused on test design, because that's really where the most of the issues that with reliability will happen are in that area. It's worth a total of 39 points. So what we did is we went through this process and gave a score, and then based on that score, we were able to give a final reliability score. And what we found is that actually none of the studies published to date are suitable for use in an environmental risk assessment of coral and UV filters. On the other hand, you had 3 that could be used as like a secondary line of evidence or within a weight of evidence, but they'd need to be repeated prior to be used as like the main key evidence within an environmental risk assessment. So they couldn't drive a risk assessment. And then finally, the rest of the 5 studies we found actually weren't suitable for risk assessment at all. And what's important to note is that the Downs 2016 study, as well as the Dan Navarro 2008 study, those were actually used to justify the UV filter bans in Hawaii. And as we found here, they actually didn't generate suitable endpoints that are reliable enough to be used in an environmental risk assessment. So this is a big data gap we have. There are currently no studies that we can use to conduct an environmental risk assessment for coral and UV filters. So up until this point, I've just been talking about organic UV filters, but I think it is important to also address what's going on in the space of inorganic UV filters. So for inorganic UV filters, we have even more limited data available. For titanium oxide, there's only been 2 studies conducted to date, and for zinc, also only 2 studies conducted to date. You'll notice that failed 2019 study also included earlier has organic UV filters in it as well. And on top of the limited hazard data, there's also very limited exposure data. In fact, there's no reef monitoring studies that have been able to be identified. And that could be because, firstly, nanoparticles are really difficult to monitor. So in some cases, like a proxy has been used to detect them. There is one study from a beach in France where they monitored directly at the beach and then a little further out into the bathing zone and then beyond the bathing zone. And what they did find is that the concentrations were decreasing from shoreline. Um, but in general, data for inorganic UV filters is even more limited than for organic UV filters. But we are currently working on environmental safety assessments for the inorganics as well. In this talk so far, I've been discussing how we can conduct an environmental safety assessment using the environmental risk assessment approach. But I also want to talk briefly about a different approach that maybe we could explore using called eco-epidemiology that might be able to, in the short term, overcome some of the data gaps that we have with the environmental risk assessments. So here I have a diagram of some of the main threats to coral reefs. So we have things like warming oceans, sea level rise, invasive species, overfishing, chemical pollutants. So within an eco-epidemiology approach, what we're effectively trying to do is ask the coral what is causing coral reef decline based on all of these stressors that we know about. So how can we do that? Well, in Hawaii, you can get data for these stressors. So you can get data on tourism, temperature, sea temperature, fishing data, pollution data, UV filter concentration data. And you can also get data on coral health from Hawaii. And then you can use a series of quite complex statistics to come out with what your main factors driving coral health are. And in theory, you could use these main factors to help drive reef management. So there has been one of these eco-epidemiology studies conducted, and the manuscript is currently in preparation. But they went out and they collected a ton of data and were able to analyze these complex statistics, and basically were able to relate key factors to the 2 key coral health indicators. In this case, it was coral diversity and coral cover. And what they found is that in terms of diversity, wave power and temperature are the big factors. Whereas for coral cover, the long-term temperature and the location in terms of latitude are really important. But the big takeaway was that based on the current data set, the UV filter concentrations aren't a key driver of coral health. It's largely driven by the sea temperature, the wave power, and the geography. So in conclusion, environmental safety concerns over UV filters have led to key scientific questions being posed. There's been a lot of studies that have been published since this concern started, and we expect there will be many more to come. Marine environments are definitely exposed to UV filters, and we are in the process of conducting environmental risk assessments to establish or evaluate their safety. But like we've just gone through, there is limited data available, and the data that is available really isn't that suitable. So it's really important to prioritize generating reliable data so we can have more conclusive assessments conducted for these UV filters. So in terms of ongoing work we're doing, we are in the process of generating a reliable test system for adult coral that is in development and conducting robust and representative UV filter monitoring in beach environments. And we're also in the process of using that data to help us to develop predictive models to actually predict the concentrations of UV filters that will be reaching coral. So taken together, we are doing our best to address these data gaps, but the science takes a long time. As I said, there's no standard test for coral, so we are in the process of trying to develop a reliable one that we can use for risk assessment going forward. And with that, I'd like to thank you all for tuning into my presentation. And if you have any questions about anything that was presented today or any ongoing work we're doing, I encourage you to reach out and drop me an email. I'd be happy to chat about it. It's been a pleasure to participate in the summit, and I thank you all for tuning in.